The ESRF, European Synchrotron Radiation Facility, is placed in Grenoble and is one of the world's largest synchrotron science centers. Its extremely brilliant X-ray for leading-edge research is used for fundamental and applied research, and often addresses how to better use the resources, develop more sustainable materials, or improve our daily lives. Every year, experiments take place at ESRF, where 9,000 scientists from all around the world, from very different disciplines such as physics, biology, engineering, chemistry, and archaeology, travel to Grenoble to expand and improve their research. Today we are with Dr. Marine Cotte of the European Synchrotron Radiation Facility of Grenoble. So, Dr. Cotte, what is your role at the ESRF?
So I have several roles. The main one is to be responsible for the ID21 beamline. This is a beamline which is dedicated to X-ray microscopy. So I have in charge the management of a team, the organization of the operation, in particular welcoming users, also the supervision of the development of the instruments, respecting the budget, trying to get some fundings. Recently, I have been appointed also as group head for the X-ray nanoprobe group, meaning that I am also, let's say, supervisor of 4 beamline responsibles, all dedicated to nanoprobe and an additional unit. In addition to that, I also push beyond ID21 and the X-ray nanoprobe group activities at the SRF in the field of heritage science. So I have been part of, let's say, collaboration to implement a new access models, which is named BAG for block allocation group, such that it's a way to give easy access and regular access to researchers in the field of cultural heritage who would like to exploit diffraction techniques. This at ID13 and ID22. And on top of that, when I have time, I try to develop my own research program in the field of cultural heritage. In particular, I have a PhD student who is working on understanding the interaction between dryers and binders, both in historical and industrial paintings. And this is in collaboration with the Rijksmuseum, University of Amsterdam, and the company AkzoNobel in the Netherlands.
Okay, and how do you discover the presence of lead calcium phosphate in artistic materials?
So the first time we detected lead calcium phosphate, it was when we were studying inks in ancient papyri from Egypt. This was through a collaboration with colleagues from the University of Copenhagen. There is a very, very important collection of papyrus, which is named Carlsberg collection. And the colleagues, they already performed analysis with their instruments, and they detected, for example, in the red, the presence of iron, and in some red and black inks, they detected lead. And it was very surprising, and they wanted to know which kind of lead was present, and if this was for which reason lead had been introduced in the inks. So they brought a couple of samples here, and we started some analysis. The first technique we used was micro-X-ray fluorescence, which is a very good technique when you want to know the elemental composition and the way the elements are distributed over heterogeneous sample. And we confirmed the presence of iron in some red inks, most of them, and also the presence of lead in black and red inks. And more than that, we revealed a very interesting distribution of lead, both at the millimeter scale and at the micron scale. And in some cases, lead was associated with chlorine, and it was clearly identified as a degradation product. But in other cases, lead was found to be co-localized with phosphorus and possibly with calcium, even if in this case the co-localization with calcium was not easy to detect because you have a lot of calcium in the papyrus. So we needed additional analysis to get a better idea about the composition of this lead compound. So we used different techniques. We used infrared spectroscopy, which was good in revealing the presence of lead carboxylates, lead salts, but failed in identifying possible interaction between lead and phosphorus. So we used another technique, which is micro X-ray diffraction, which is very good when you have crystalline phases that you want to identify and locate. And surprisingly, we detected lead calcium phosphate. And also, by pure serendipity, almost at the same time, we found similar phases in historical paintings. So then we decided that since there were not so many records about this kind of lead calcium phosphates in the heritage domain, except in the few cases of corrosion where they have been associated with some degradation, we saw that it could be good to launch a project on that and to see how the lead phosphates form and what are the reasons for their presence in some historical materials.
So, what is a synchrotron and how it works?
So here we are at the European Synchrotron Radiation Facility. Seen from the sky, basically, the synchrotron is a kind of ring. It's a huge ring with a circumference of about 840 meters, so it's a huge facility. At the core of this ring, you will find an accelerator, which is producing and accelerating a beam of electrons. These electrons, once they are sufficiently accelerated, they are sent into the storage ring. In the storage ring, under certain conditions, when the electrons are submitted to some magnets, you produce what we call the synchrotron light. This synchrotron light has many properties, and for what concerns the SRF, this is mainly produced in the domain of X-rays. So you can consider that the ESRF is a kind of extremely bright source of X-rays. When these X-rays are produced, they go straight tangentially to the synchrotron ring. And so around the ring, you have about 44 what we call beamlines. And each beamline, as we are here at the ID21 beamline, is a kind of separate laboratory. So each beamline will exploit X-rays differently. We have our own instruments, we have our own detectors, we have our own sample environment or sample stage. Here, for example, we are specialized in X-ray microscopy, meaning that we will exploit the capability to produce very small beam to get insight into the matter with a very high resolution. Also, the energy of the X-rays will give the specificity of beamlines. Here we are in a beamline which is what we call tender X-ray domain, meaning it's not super high energies, but there are other beamlines at the European Synchrotron Radiation Facility, which are optimized for high energies X-rays. Other specificity of beamlines are, for example, the possibility to work under cryo conditions. So when you work on biological samples, you want to reduce the risk of damage on your sample, and being in cryo conditions is one of the options. Also, the detectors or the detecting geometries that you will implement will completely define one beamline with respect to another. We all use X-rays, but we use different techniques, and so it means that we can provide users or researchers with different and complementary information about their samples.
And why is it so important? What are the gains over the conventional X-ray techniques?
So there are many properties of the synchrotron X-ray beam, in particular the brightness. The brightness is basically the number of photons emitted per unit of surface, the unit of solid angle, unit of spectral bandwidth, and unit of time. So briefly, it means that you have a lot of photons arriving shortly in a small area. So you can have experiments that you could also have in the laboratory, but much faster and with much better quality. On top of that, you have the fact that the energy of X-rays can be tuned and selected such that we can perform spectroscopy, which is not something you can perform easily with standard X-ray sources. An additional aspect is coherence. So it's a unique property of a synchrotron beam, and which can be exploited to a technique which is named phase contrast tomography. So there are many aspects of the synchrotron beam which can be exploited into let's say, outperforming techniques that you can find in the lab, and also proposing new techniques which do not exist with standard laboratory equipments.
And how can a synchrotron analysis help the heritage science?
So there are many ways synchrotron facilities or synchrotron techniques can be used in the field of heritage science. You have techniques or typically phase contrast tomography, which will be used a lot in paleontology, where you want basically to understand the origin of life. For example, the evolution between species, the evolution from a child to adult, all possible, let's say, natural evolutions, which will translate in some specific structures of the living materials. And so this tomography technique is used a lot in paleontology to get insight into the internal structure of the material. And you have other applications which are more based on chemistry and more related to cultural heritage. So in this case, you have two main questions. Questions related to how the objects were produced. So if people were using, or craftsmen were using, natural or synthetic materials, under which conditions like temperature or reductive oxidative conditions, ceramics or glasses were fired. How a painter developed specific techniques. All these questions where chemistry can give you information about artistic procedures. And on the other hand, you have experiments dedicated to the conservation of artworks. So understanding the composition of degradation products, assessing which factor, internal or external to the objects, are responsible for the degradation, assessing as well conservation treatments. So there are many ways where synchrotron techniques can be used to get insight into objects and preserving them for the future generations.
Today, we just had an example of what synchrotron analysis can be useful to, its characteristic and its application. But if what you just watched today can be the trigger for a new scientific research, how is it arranged? How can chemistry help to solve this question? Which are the following steps? Well, in the next episode, we will try to reply to this and other questions, and to get you more into heritage science. So, see you next time.